What is cancer? Cancer is a group of diseases characterized by uncontrolled, abnormal cell division due to genetic mutations that disrupt normal cell cycle regulation. These abnormal cells can invade nearby tissues, invasion, spread to distant organs via blood or lymph, metastasis. Key feature: loss of control over cell growth, differentiation, and apoptosis, programmed cell death. How many types of cancer exist in humans? Over 200 different types of cancers are identified in humans. Classification is mainly based on cell/tissue of origin, molecular and genetic features. Major types of cancer, based on tissue of origin. A. Carcinomas, 80 to 90%. Origin, epithelial cells. Examples: lung cancer, breast cancer, colon cancer, prostate cancer, skin cancer, basal cell, squamous cell. B. Sarcomas. Origin, connective tissues. Examples: osteosarcoma, bone, chondrosarcoma, cartilage, liposarcoma, fat, leiomyosarcoma, smooth muscle. C. Leukemias. Origin, bone marrow and blood-forming tissues. Features: no solid tumor, excess abnormal white blood cells. Examples: acute lymphoblastic leukemia, ALL, chronic myeloid leukemia, CML. D. Lymphomas. Origin, lymphatic system. Examples: Hodgkin's lymphoma, non-Hodgkin's lymphoma, myeloma. Origin, plasma cells. Example: multiple myeloma, melanoma. Origin, melanocytes, pigment cells. Highly aggressive skin cancer. G. Germ cell tumors. Origin, reproductive cells. Examples: testicular cancer, ovarian cancer. H. Central nervous system, CNS, tumors. Origin, brain and spinal cord cells. Examples: glioma, astrocytoma, medulloblastoma. Process of cancer occurrence and development, carcinogenesis. Carcinogenesis is a multi-step process involving genetic and epigenetic changes. Step 1. Initiation. Permanent DNA mutation occurs, caused by carcinogens, chemicals, radiation, viruses. Mutation alone is not sufficient for cancer. Step 2. Promotion. Mutated cells receive growth signals. Increase cell proliferation. Changes are reversible. Step 3. Progression. Accumulation of multiple mutations. Cells become highly proliferative, invasive, resistant to apoptosis, leads to malignant tumor formation. Step 4. Metastasis. Cancer cells detach from primary tumor, enter blood/lymph, colonize distant organs, lung, liver, bone, brain. Molecular basis of cancer. A. Oncogenes. Mutated proto-oncogenes. Promote uncontrolled cell division. Examples: RAS, MYC, HER2. B. Tumor suppressor genes. Normally inhibit cell growth. Loss of function leads to cancer. Examples: P53, guardian of genome. RB, BRCA1/BRCA2. C. Defective DNA repair genes. Allow accumulation of mutations. Examples: mismatch repair genes, MLH1, MSH2. Factors responsible for carcinogenesis. Aphysical factors: ionizing radiation, X-rays, gamma rays. UV radiation, skin cancer. B. Chemical factors: tobacco smoke, benzopyrene. Asbestos. Aflatoxins. Industrial chemicals. C. Biological factors: viruses. HPV may cause cervical cancer. HCV may cause liver cancer. EBV may cause lymphoma. Bacteria. Helicobacter pylori may cause stomach cancer. Parasites. Schistosoma may cause bladder cancer. D. Genetic factors: inherited mutations. Family history of cancer. E. Lifestyle factors: smoking, alcohol consumption, poor diet, obesity, lack of physical activity. F. Hormonal factors: estrogen may induce breast and endometrial cancer. Androgens may induce prostate cancer. Hallmarks of cancer: cancer cells acquire abilities such as sustained proliferative signaling, evading growth suppressors, resisting apoptosis, enabling replicative immortality, inducing angiogenesis, activating invasion and metastasis, genome instability, immune evasion. Treatment of cancer: A. Surgery. Removal of solid tumors. Most effective in early stages. B. Radiation therapy. DNA damage using high-energy radiation. Localized treatment. C. Chemotherapy. Cytotoxic drugs targeting rapidly dividing cells. Examples: cisplatin, doxorubicin, methotrexate. D. Targeted therapy. Acts on specific molecular targets. Examples: imatinib, BCR-ABL, trastuzumab, HER2. E. Immunotherapy. Boosts immune system. Examples: checkpoint inhibitors, PD-1, CTLA-4. CAR T-cell therapy. F. Hormone therapy. Used in hormone-dependent cancers. Examples: tamoxifen, breast cancer. Androgen deprivation therapy, prostate cancer. Gene and cell-based therapy, emerging. CRISPR-based therapies. Cancer vaccines. Personalized medicine. Conclusion: cancer is a complex, multi-step genetic disease involving uncontrolled cell growth, influenced by environmental, genetic, and lifestyle factors, and treated using surgery, drugs, radiation, immunotherapy, and advanced molecular approaches. Thanks for watching.